Welcome to the Cell Metabolism Lab, where I've prepared 1, 2, 3, 4, 5, 6, 7 beakers and Erlenmeyer flasks. Okay, so here is what we've got. I know you can read it. We've got a container that contains live yeast. We've got a container of just water. We've got a container of 5% sucrose. That's an real sugar, that's table sugar. And then we've got 3 artificial sweeteners, 5% of equal, 5% of Splenda, and a 5% solution of Truvia. And then, in our last little jar, we've got some very unhappy dead yeast. Poor little guys. Okay, and our first jar has got live yeast. They're happy. Okay, so what we want to do is, since we don't have human cells that we can work with and measure their cell respiration, and when our cells respire, they produce a little CO2 waste and they make some energy. Well, we don't have human cells, but we do have live yeast cells, and yeast can ferment anaerobically sugar, and they will produce CO2 as a byproduct as well. So we've got sucrose, which we know yeast can metabolize because we have a wonderful beer and wine industry that uses mixing live yeast with sugars to make alcohol. But will yeast recognize artificial sweeteners? We're going to find out. Okay, now here is our experimental setup. I'm going to make 1, 2, 3, 4, 5, 6 experimental groups. And it's simply going to be, I'm going to take little test tubes and fill them. So I'm going to fill one test tube with 2 ml of live yeast and 2 ml of plain water right out of the tap. Next, I'm going to make a solution of 2 ml of live yeast and 2 ml of 5% sucrose. And then I'll keep going, adding 2 ml of live yeast. Next, I'll make up 2 ml of 5% equal, 2 ml of live yeast, 2 ml of 5% Splenda, 2 ml live yeast, 2 ml 5% Truvia, and lastly 2 ml of dead yeast with 2 ml sucrose. Okay, so the ones marked with the minus sign are negative controls because, well, we expect those not to work. And, well, water doesn't contain any sugar, so what would yeast have to metabolize? The dead yeast, dead things don't metabolize. So even though it's got sucrose, dead things can't metabolize and produce anything. We've got a positive control, which is your table sugar. Like I said, we know yeast can metabolize table sugar. So here's what we're going to do. We're going to move over to the pipette bench. I'm going to put a pipette on. It's a 1,000 microliter pipette. That's 1 ml. And to that, I'm going to add, here's 1 ml of my live yeast. Here is another ml. Awesome. Okay, and then I'm going to make up the sucrose solution. So I'm going to put 1 ml of sucrose, the table sugar, the real sugar. Okay, we are ready to go. We're going to mix, 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 and then we're going to put it into a water bath that is set to 37 degrees. And we're going to set our timer for 5 minutes. Fooled ya, time warp. I've already made some up. Okay, this has come out of the incubator after 5 minutes, and I'm going to put it into the respiration chamber, pour it all in. Okay, and now I'm going to use a CO2 probe to measure the CO2 produced. So I'm going to put that on, and here is our Logger Pro program that is going to measure CO2. So I'm going to hit collect. And what will happen is every 30 seconds, that probe is going to measure CO2. There's our first reading. And so we're going to come back in just a minute and see what has happened with our CO production over 5 minutes. All right, our yeast and sucrose solution is finished running. We've got that 300 seconds of time and it cut off on its own. We've got CO2 readings, and now what we're going to do is we have to calculate delta CO2. Delta CO2 is simply the difference between the lowest CO2 reading and the highest. So my highest CO2 reading was 5,283. My lowest CO2 reading was 653. So, go back to my calculator. 5,283 minus 653. My delta CO2 equals 4,630. Okay.